Many of you already know him. He is an ND PhD student in Dr. Simon Schenk laboratories. Leon's interest in biomedical research actually started like a long time ago. If you think about it, it started in 2016 when he participated in the Ragon Institute in Mass General Hospital, MIT, and Harvard undergraduate summer internship program, where he was working in the lab of Dr. Sylvie Legal on projects related to HIV. And 3 years later, he graduated in biochemistry and molecular biology at the University of Massachusetts Commonwealth Honors College, where he received as well the undergraduate research fellowship to conduct research in the lab of Dr. Peter Chien in the Department of Biochemistry and Molecular Biology. And in the Chien's lab, Leon studied something that I just discovered, that he was working in protein degradation and quality control in Caulobacter crescentus, which is a very interesting gram-negative bacteria. Leon has also completed like a 6-month internship in a company in Sanofi Genzyme. So he has also some hint of how doing research in a company is. Here at UC San Diego, he completed 2 years of medical school, and currently he's in the biomedical science PhD program. He has 2 Astros accepted by the Clinical Orthopaedic Society and the Eastern Orthopaedic Association. He won the Society Hospitals Medicine Research Innovations and Clinical Vignettes poster competition, and he presented at the annual research conference in 2021 in Nashville. He was awarded a summer research training program grant from the UC San Diego School of Medicine, and he conducted summer research under the guidance of Dr. Schenk. He discovered his passion about exercise and muscle biology. So he decided to stay in his lab and move forward with his dissertation. So today he's going to talk about the time of day variability in exercise performance. He will talk about a deeper look into the role of the skeletal muscle. So thank you, Liam, and we are looking forward to hearing from you.
Awesome. Thank you for the introduction. I'll just share my screen here. All right. Do you guys see just the presentation? Yeah. Cool. All right. So let me just get my laser pointer up here. Boom. So good morning, everybody. Once again, thank you for the introduction. My name's Liam, and today I'm going to be talking about time of day variability in exercise performance and taking a deeper look into the role specifically of skeletal muscle. So during this talk, I'm first going to give a little bit of background on the molecular basis of circadian rhythms and kind of summarize the state of the field as it relates to chronobiology and the effects of time of day on athletic performance. I'll then talk about the actual experiments that I did, looking at the impact of time of day on skeletal muscle contractile function and contraction-stimulated glucose uptake. I'll show some of my data and then get into kind of my main takeaways at the end to wrap it all up. And so, like I mentioned, we'll start with kind of a little bit of a summary of the molecular basis of circadian rhythms. And so going back as far back as I could in the literature, the first scientific writing that we have about circadian rhythms comes from this journal. I think it translates to the French Academy of Sciences. It's written by this scientist at the time, Jean-Jacques something. I'm not even gonna attempt to pronounce the rest of his name, but I'll just call him JJ, I guess, from now on. But so this guy, Jean-Jacques, he wrote the first article published in a scientific journal related to circadian biology. And in this article, he makes this interesting observation that the Mimosa pudica plant opens and closes its leaves, depending on the time of the day. So when the sun rises, the leaves open up, and when the sun sets, the leaves close again. I don't know if anybody's ever seen this plant, in the wild before. It's actually cool because it not only opens and closes in response to sunlight, it also closes if you touch it. So it's something just cool to play with if you see it around. And it happens very quickly too. But he was interested in the opening and closing based off of time of day. And he wanted to test whether or not the plant is just sensing the sunlight and there's, something going on where it's causing, detecting metabolite and photosynthesis or something, and that's what's causing it to open and close, or whether the plant has its own endogenous clock where it's able to keep time on its own in the absence of the sun. So what he did was he took the plant and he left it out in the sunlight, for a couple of days or however long. So it was trained and had that rhythm where it was opening and closing to the sunlight. And then he moved it to his basement where there was no sunlight. And he noticed and observed that the plant continued to open and close its leaves even when it's in the basement. And interestingly enough, he says that this might have a probable connection with the ability of a lot of sick people to differentiate day and night from their bed. Something that I think all of us can relate to. I mean, if you've ever set your alarm clock for 7 a.m. and woken up at 6:55, I think you have this sense that we have some kind of internal timekeeper um, in our bodies. So this is kind of a one way that he went about testing that. And so since this is the first documented instance of a endogenous basis of the circadian rhythm, and basically 300 years passed before we made any more headway in what the actual molecular basis is. And like a lot of things, when you're trying to find out the molecular basis of something, a lot of it was figured out using flies in the 70s. Seems like that's where a lot of the molecular everything comes from. But anyways, yeah, so it was the first genetic basis for circadian rhythms was discovered in 1971. And so for this experiment, they used Drosophila, so houseflies, and Drosophila have their eggs hatch in a cyclic fashion that goes with time of day. So this is a figure here showing the eclosion or egg laying rhythms of a Drosophila. And so you can see that the eggs hatch pretty much at the same time of day every day in a periodic 24-hour cycle. And so what they did was they looked at thousands and thousands of egg laying and egg hatching or egg laying events, and looked at when the events happened within the bell curve, whether the egg laying happened earlier or whether the egg laying happened later. And after doing this thousands and thousands of times, I think in the paper they said they did it over 2,000. They looked at over 2,000 egg laying events that identified three different phenotypes beyond the normal eclosion rhythm. So these are the three different phenotypes that they identified. They identified an arrhythmic phenotype, where you can see instead of these nice peaks and valleys on a 24-hour cycle, the eclosion events are kind of happening randomly over time. They have a short period. So instead of laying eggs every 24 hours or so, laying eggs every like 20 to 22 hours. Then they have a long period where instead of 24 hours, it's, 25, 26, 27 hours between eclosion events. And interestingly enough, they noticed that these strains of flies don't only have aberrant eclosion rhythms, it also translates into the locomotor activity of the fly in adulthood. So they basically put the flies in a box and they're trained to a 12-hour lights on, 12-hour lights off rhythm for a couple of weeks. And they shut the lights off and measure how many times the flies basically bang into the glass on the side of the cage. And that translates to the fly's locomotor activity. And as you can see that whether it's the arrhythmic short period or long period, these effects that you see or this phenotype that you see for the eclosion rhythms translates to their activity in adulthood. And after doing genetic analysis of each of the different strains, they were able to isolate each different phenotype being due to a different mutation on a gene in the X chromosome, which they called period. So this is the first, in period, as we'll find out in a little bit, ends up being one of the master regulators of the circadian clock. But this is the first time that gene was reported on. A couple of decades passed and people started to figure out how the actual circadian loop oscillates on a 24-hour cycle. So this paper came out in 1990, and it's the first hypothesis and first direct evidence that it could be due to a transcription translation feedback loop. And this piqued a lot of people's interest. And basically over the next decade, people had fully flushed out the way the circadian cycle works. So by around the turn of the millennium, the molecular basis of the circadian clock had been fully established. And so kind of a quick summary of the circadian clock. Almost all tissue in your body has its own circadian clock, but the master circadian clock that informs these other clocks is located in the suprachiasmatic nucleus of the hypothalamus. And this clock is tuned by lots of different things, by meal timing, most primarily tuned by light, which I don't think is surprising to anybody. But so there's this clock in your brain, and it has a molecular basis that helps it tick on a 24-hour cycle. And so, as I mentioned earlier, it's via a transcription translation feedback loop. So basically, you have these proteins, BMAL1 and clock, which are transcription factors for other genes, CRY and PER, REV-ERB and ROR, and they cause the transcription of mRNA from these genes. Those are translated into, for example, CRY and PER proteins. However, those proteins are inhibitors for BMAL1 and clock. So what happens is you have BMAL1 and clock around. They cause the transcription translation of CRY and PER, for example. And then CRY and PER inhibit BMAL1 and clock. So if you're inhibiting your transcription factor, these protein levels are going to go down. You'll relieve the transcription, and now you'll have BMAL1 and clock around again. And this happens in a cyclical fashion over approximately 24 hours, which is how the circadian cycle is tuned within our brain. And this same clock mechanism is present in basically all of our peripheral tissues, whether it's adipose tissue, liver, heart, skeletal muscle. So this is the molecular basis of the circadian clock. And besides just the molecular basis, I think we all have an understanding of how much our physiology changes based off of time of day. So circadian rhythms can impact all different aspects of our physiology. So I think most obviously it impacts our sleep-wake cycle. It also impacts body temperature, blood pressure, your appetite, hormonal secretion, and your activity levels throughout the day as well. And so it impacts all these different aspects of physiology. And a question that people have had for the past 60 years or so is, how does it impact athletic performance? Because physiology informs how you perform in sports. And so you would imagine that if your physiology is changing depending on the time of day, there could potentially be impacts on athletic performance. And so scientists started first looking at this in the late 60s, early 70s. This is the earliest paper I could find where they looked at 10 women and 20 men from a university, 16 swimmers, 6 runners, 3 shot putters, and a rowing 4, plus a coxswain. And they had them do their respective events between the hours of 7 and 8 in the morning and 5 and 9 p.m. on the same day for each group. And overall, they saw that basically all of the athletes did better in the evening than in the morning. And this is just kind of a observation that they had, but this is the first piece of literature I could find where people are looking at the effects of time of day on athletic performance. And this trend of people performing better in the evening actually ends up being observed in more controlled experimental studies decades later, which I will be getting into. But a lot of the evidence.
Liam, I had a quick question on that. Just this is mostly, I mean, obviously this is not your work, and you've read more literature since then. But I would just note that it does say that 6 of the swimmers who, I guess, typically worked in the morning did worse in the evening. So there's a lot of variability in this. Even though the runners improved, the swimmers did not necessarily improve. So it's not cut and dry.
It is certainly not cut and dry, and that is an issue that I have that I will be getting into in some detail later for sure. And so a lot of the evidence for time of day effects on sports performance is after the fact evidence. So kind of retrospectively looking back at performance in different events at different times of day and saying, for example, oh, if you look at every world record that's broken in sports, pretty much, the vast majority of them happen in the evening as opposed to the morning. And you could say, oh, that's because your circadian cycle is more attuned to, your body is more optimized and primed for working out at this phase of the circadian cycle. But there are many other factors to consider. I mean, the training effect, have you had time to practice that morning? Temperature, it typically is warmer towards the afternoon and evening than first thing in the morning. And we know that temperature can improve athletic performance. Having it be a little bit warmer out as opposed to being a little bit colder can definitely have an effect. And then specifically with world records, for example, there's world records are typically aired, events where a world record could be broken are typically aired on television, which has an incentive to show the program to as many people as possible. So these are usually prime time evening slots. So there's a lot of bias and things that can confound this type of data. There's another study I found that I think is actually pretty interesting that looks at the effect of jet lag on team performance in the NBA. So in this study, they looked at 10 seasons worth of data. It was like over 11,000 games that they looked at. And they looked at teams flying eastward across time zones versus westward across time zones. And when they do this, and compare that to teams that are traveling within their time zone for a game or have a home game. And what they found is that when traveling westward, there was no significant difference in winning percentage. However, when traveling eastward, there was a significant drop in winning percentage. They also found that there was a significant drop only when traveling eastward in points differential as well. So there seems to be this effect of jet lag, which people will say is, oh, because you're messing up your circadian cycle. So that's why you're seeing this effect. But in my opinion, if this were true, you would see an effect going both eastward or westward. And I think, in my opinion, this comes down to traveling eastward, you get a couple hours less of sleep that night. whereas when you're traveling westward, you gain a couple hours of sleep. So I think it has to do more with recovery, and having more time after travel to, a long travel day to sleep and recover, as opposed to losing a couple hours of sleep when you're traveling eastward. And just interestingly enough, too, when you look at the magnitude of traveling eastward, so doing a 1 hour time difference eastward versus a 2 hour time difference eastward, there's kind of a dose response effect, which I think is pretty interesting. I didn't include this data on the slide, but in the paper, they basically conclude that people, the reason for the points differential and the difference in loss is that players aren't going for rebounds as often during the game, which in my opinion has to do with just being less rested. So pretty interesting paper, but in my opinion, not direct evidence of the circadian rhythm effect on athletic performance. So those are kind of retrospective studies. Looking at more direct studies where we're actually looking at athletes within a day doing a standardized test, there is a pretty large amount of studies showing this effect of time of day, where people tend to be better in the evening versus in the morning. So like in that first paper from 1973, none of the swimmers showed any effect. But in this paper in 1983, they showed a pretty robust effect of time of day on swimming. So they had 14 competitive swimmers, 4 male, 10 female. Median age was 14.7. And they came in at 5 different times of day. And they did an all-out 100-meter and all-out 400-meter swimming test. And I just showed the 100-meter here because the graph basically looks the same for the 400-meter. But there's this pretty substantial improvement in a 100-meter test based off of time of day. I mean, this is 2.5 seconds, which is, in the world of competitive swimming, is an eternity. So they demonstrated an effect here, but again, no real explanation as to why. But this is just an issue that I have with the field in general of circadian biology, is there's a lot of, we observed this, it's different at a time of day, so it has to be due to the circadian cycle. And even small things like in the paper, they do body temperature and show that body temperature is substantially higher towards the end of the day, which has a correlation with exercise performance. And this trend of body temperature being higher towards the end of the day, this trend is seen in basically every study of athletic performance based off of time of day. So it's very difficult to disentangle those two variables. Some other data that we have on athletic performance is looking at tennis serve velocity and accuracy. And interestingly enough, they show that as the day goes on, so we have 9 a.m., 2 p.m., 6 p.m., that the 9 a.m. Serve velocity is significantly lower than all the others. However, it's also significantly less accurate than the others. So suggesting some kind of time of day effect on velocity and accuracy. However, this is just 6 tennis players, on a 1-day, one-off study. And in my opinion, this could, at least what we're seeing with serve velocity, could just be due to a training effect where they've had, they're kind of getting the rust off in the morning. They haven't practiced at all. And then, at the second and third times of day that they're testing, they'll already have served earlier in the day. So in my opinion, I would expect them to perform better, which means that it doesn't necessarily have to be a circadian effect. It could be due to the training effect, or body temperature, or ambient temperature outside, which wasn't measured, or meal timing. Maybe they had a bunch of carbs right after breakfast and now their glycogen is higher here than it was in the morning. They didn't control for whether they had eaten breakfast before they came in or not. So I think you have to take a lot of these studies with a grain of salt. People have also looked at mobility. So whether it's glenohumeral lateral rotation or whether it's a fingertip to floor measurement, people have documented an increase in flexibility that tends to peak in the afternoon to evening. And they've looked at strength, so doing a leg extension and leg flexion measurements and looking at peak torque. You see a trend towards higher torques towards the end of the day versus earlier in the day. And then not only strength, but people have done this with power as well. And I think this study is interesting because they're looking at trained individuals. So they had 14 subjects, all male, come in and do a 16-week training regimen. I mean, a 6-week training regimen, excuse me, of like extensions. And after the 6 weeks of training, come in and do a Wingate test and looking at anaerobic power. And what they see is, so the circles and the squares, one thing that's interesting about this study as well is they had people train only in the morning or train only in the evening. So they separated them to 2 different groups of morning training, evening training, had them come in only in the morning for those 6 weeks or only in the evening for those 6 weeks, and then come back and repeat the study. So as you can see, both groups pre-training have start off at this certain level of anaerobic power in the morning. When they come back in the evening, both groups perform better and produce more power on the test. And then interestingly enough, after the 6 weeks of training, you can see that the morning training group performs better and performs equally the same in both the morning and the evening. Whereas the evening group performs better in the evening, they don't perform better in the morning. So I think this is interesting because there's all of this data about, oh, you perform better in the evening. So, you only have to train in the evening or should only train in the evening. Or I think it should almost be the opposite. Whereas if you train better in the evening and your morning is your weakest time of day, I think this is some evidence that by specifically training in the morning, you can overcome that disadvantage and learn to become a good morning athlete versus only good in the evening athlete. So it is trainable. And this is what they like to call your chronotype. So kind of like a night owl versus morning bird type of personality. You can train yourself out of it, which I think is interesting. And so besides human studies, there have been several studies looking at mice. So this is one study, for example, and there's a lot of studies like this where they do a test of exercise, typically endurance. So whether it's a treadmill running test or a swimming test, and then they do a bunch of omics types measurements, so metabolomics, transcriptomics, proteomics, and show based off of time of day, these mice are performing differently on this exercise task. And based off of time of day, we see these big shifts in, for example, the transcriptional or metabolic landscape of the cell. So inside the muscle. So they say we have difference in transcriptional landscape inside the muscle, or difference in metabolic landscape inside the muscle based off of time of day, difference in performance based off of time of day. So that means the difference in performance that we're seeing is due to the muscle, which I think for reasons that I'll get into in a little bit is not a correct leap that you can make. And so this is just an example of some of that type of data where they do a treadmill running test on mice, and they showed the mice that do the test earlier in the active phase. So because mice are nocturnal, they're active at night. So this is like 2 hours into nighttime for the mouse is the early phase versus late, which is about 2 hours before the lights turn on. So that towards the end of the mouse's active phase, that the mouse runs for significantly longer on the treadmill. However, as you can see in the measurement of blood glucose, the early versus the late group has significant differences in maintenance of blood glucose throughout the exercise. And in the paper, the authors say that there was no significant difference in food consumption before exercise for the two different groups. However, if you actually look at the figure, even though they say there's no significant difference, the early versus the late group, it's like a 40% difference between the groups. So maybe it's not significant, but in my opinion, that's just means that it's underpowered because that we're talking about, almost half a gram of food. And if a mouse is only eating, 4 to 5 grams of food a day, that's a significant amount of food. And this is just in the 2 hours before the experiment. So this is food that's going, into the bloodstream in the kind of acute period right before the exercise. So it does matter. And so out of kind of frustration with lots of the experiments, both in humans, the retrospective studies looking at professional sports, looking at these mouse studies, there are just a lot of leaps and jumps being made that skeletal muscle is the reason why we're seeing these effects in the circadian rhythm of the circadian rhythm of the skeletal muscle is why we see these differences in athletic performance based off of time of day. And as I mentioned earlier, I just don't think that is an appropriate leap because, as I mentioned earlier as well, there are all sorts of things that vary based off of time of day, body temperature, perception of fatigue or alertness, pain threshold has been shown to vary throughout time of day. VO2 can change based off of time of day. There are all sorts of different things, and that's just within your body. Then there's all sorts of other variables like ambient temperature. What is the last time you ate a meal? What was the meal composition? Did you eat a ton of carbs the night before? And are you trained versus untrained? So there are a lot of things that aren't being controlled for in these studies. So our goal with the study that I did was to control for as many variables as we possibly could and take a magnifying glass and just really zoom in on the role of skeletal muscle itself and not disregarding, but trying to control for everything else that we possibly could. So for this study, we did measurements at 4 different times of day. So in the world of chronobiology, everything goes in Zeitgeber time. So ZT0 is when the lights turn on in the vivarium. ZT12 is when the lights turn off. And then at ZT24/0, the lights turn back on again. So everything in this, the data that I show will be in Zeitgeber time. So the 4 times that we did were Zeitgeber time 1, 7, 13, and 19, which correspond to 7 a.m., 1 p.m., 7 p.m., and 1 a.m. And at each of these 4 time points, we took a mouse and 3 hours before the experiment, gavage it with a standardized last meal of dextrose. And so we think this is important and different from other studies done in mice and humans because most of them aren't controlling for meal timing, meal size, and meal composition, just something that we do in the study. So each mouse 3 hours before the study gets dosed with the same amount of dextrose. So they're getting the same meal every mouse every time, and then a standardized fast for 3 hours. So the mouse fasts for 3 hours before each experiment. And then after the 3-hour fast, we start the experiment. So for this example, we would gavage the mouse at 4 p.m., fast for 3 hours, and start it at 7 p.m. And we'd make sure, of course, that at 6 o'clock, the mouse would go from light to dark. So we'd put it in a light-proof box, then put that in a dark room and close the door and make sure to do everything under dim red light if it happens during the dark phase of the circadian cycle. And so when we actually do the experiment, we chose an ex vivo approach. So we remove the muscles, we surgically remove the muscles out of the mouse and stimulate the mouse with, stimulate the muscles from the mouse with electrodes. And we measure, we did a force frequency curve, so stimulating the muscle with increasing frequency and measuring the force that the muscle is able to produce. We did an endurance test where we continually and repeatedly stimulate the muscle over a 15-minute period and watch the force go down over time. And we also measured glucose uptake into the muscle, because uptake of carbohydrate into the muscle can have a huge effect on muscular performance and muscular endurance. So we measured that as well. And so this is just a picture of our ex vivo setup. So this is a mouse muscle here, and it's tied on either end to these 2 posts. This post is a fixed post and doesn't move. And this post here is a force transducer. So we have these 2 platinum electrodes on either side, and the muscle is bathed in a buffer that allows a current to be passed through the muscle. And so when we stimulate the muscle will contract, it will pull on this lever arm, and then we can measure the forces that the muscle produces as the muscle contracts. So there should be a video here. Let's see if I can get this to play. So here's the muscle. You'll see it contract on the TV in a second. So there's the contraction. And then we can look at the force tracing afterwards. So you can see if the muscle is resting, it's stimulated, it rapidly produces force. As we continue the stimulation, the force drops off over time, and then we stop the stimulation here, and the muscle goes back down to baseline. And so just a little bit of rationale about why we chose ex vivo versus in vivo muscle contractions or in vivo experiments, like for example, running on a treadmill. And I've talked about this a little bit earlier, but just to kind of summarize it all, the in vivo experiments have been poorly controlled for lots of things like temperature, meal timing, et cetera, based off of time of day. There are documented things like changes in pain perception and perceived fatigue, which can impact participation in something like an endurance test and can for sure impact the results. And, you know, these are the types of studies that people have said, oh, because there's differences in this treadmill test, it has to be due to the muscle, which I just don't think you can do that, make that assumption from a full body, more systemic test of athletic performance. Versus with ex vivo, we can control for lots of different variables. So we can control for the temperature. Everything was done in buffer of the same temperature. Oxygen delivery. Each muscle gets, we perfuse oxygen through the solution at a standardized rate for every single muscle. We're removing the effects of blood flow, humoral factors in the blood, nerve stimulation, perception of fatigue. We get around all of this by doing an ex vivo approach. And I think, interestingly, it allows us to look at muscle specific and fiber type specific effects of exercise. So every study that we did, we looked at both the extensor digitorum longus muscle, which is a more fast twitch muscle, and the soleus muscle, which is a more slow twitch muscle. So it kind of helps us look at these fiber type differences and to compare different muscles of different fiber types to each other, as opposed to just looking at the whole mouse. So to get into some of my actual data here, so the first thing that we do in the protocol is a force frequency curve. So we stimulate the muscle at increasing frequencies and then measure the stress that the muscle is able to produce, so how hard it's pulling on that lever arm. And as you can see, for all 4 times of day that we tested, as you increase frequency, there's an increase in stress produced by the muscle. So it's pulling harder and harder on the lever arm. However, it wasn't statistically different between the 4 different time points. And so this is looking at maximal stress here. So we're zooming in just on these last few, or the 120 hertz data points. You can see that depending on, no matter what time of day it is, they all are producing the same amount of maximum force. And this, we could make a graph like this for 100 hertz, for 40 hertz, for 1 hertz, and all these graphs tell the same story. And like we said, we not only did this for the fast twitch EDL muscle, we also did this for the slow twitch soleus muscle. So no matter what time of day it is, as we increase the frequency, we see an increase in stress. However, it's not dependent on time of day. So it doesn't seem like time of day impacts muscle contractile function, whether it's a simple muscle twitch or whether it's a fully maxed out muscle is being stimulated as much as it possibly can, or anywhere in between. It doesn't seem like time of day impacts muscle contractile function.
Liam, I
Yeah.
think I might miss this. These are male mice, or they are?
Oh, yeah.
You use both female and males to see whether there is a hormonal component that may affect?
That's a good question. So we actually did it in only female mice.
Okay.
These are all female mice, all roughly 13 weeks of age.
Okay. Thank
And
you.
all of them too, yeah, of course. And all of them after this is all done in C57 Black 6 mice. So we ordered them, from Jackson, and then we made sure to leave them in the vivarium for 3 weeks before doing any experiments, just to make sure that all of the circadian rhythms were synced up. Looking at the literature, seems like 2 weeks is what most people do, but we just wanted to be completely sure that that all the rhythms were the same.
And how old are they?
13 weeks. 13
Okay.
weeks. Yeah.
Thank you.
Yes, of course. And so the next thing that we looked at is muscle fatigability, or, endurance, muscular endurance. And so in this test, we stimulate the muscle every couple of seconds. And as you repeatedly stimulate the muscle, you see that it loses force over time. And we can track that and look at this kind of curve here. And so this is just looking at raw forces being produced by the muscle. And depending on time of day, there's no difference at any time point in this fatigue curve. And so this is both looking at raw forces, and if we normalize the data as a percentage of initial stress, so everything starts at 100%, and when we look at each force drop off, and instead of having a raw stress, convert it to a percentage of that initial force, the story is still the same, where depending on time of day, there's no difference in this endurance curve. And we can calculate kind of total, cumulative stress by looking at area under the curve for the fatigue curve. And there's no difference between the different time points for both EDL and for soleus. The story is the same, whether you're looking at raw stress, stress as a percentage of initial, or area under the curve. It seems like that time of day does not impact the fatigability of each muscle.
Liam, could I ask you a quick question?
Yeah.
So I know you're going to look at glucose uptake in subsequent slides. So there's a lot of variability in each of your groups, like substantial variability. Are
Mhm.
you measuring anything else besides glucose uptake that you might be able to, so take the light dark portion out of it, but just that you might be able to correlate with performance other than glucose uptake, or are you pretty much just sticking to these pretty focused set of measures?
Yeah, so for this study, this is all we've done so far. The plan is also we want to do some qPCR on a bunch of circadian regulated genes in muscle, and it'll be interesting to look at different groups and see if how those are varying based off of time of day, and then also within each of these time points, each of these specific muscles, we not only you know, we measured blood glucose, glucose uptake, and we also have serum, so we can measure things like cortisol levels, glucagon levels, insulin levels, and things like that, which I think would be interesting to look at that as well, and then see if it aligns up with if there's a trend, within a time point, for example, with those different measurements. So we have tissue banked and some things that we want to look at down the road, but this is the data that I have right now.
Cool, thanks.
Yeah, of course.
So Liam, there is a paper that we had discussed a long time ago during lab meeting where they ended up, it's completely like, they're not just looking at exercise and muscle, they were looking at nerve regeneration after injury. And they were looking at the differences between light and day. And they pretty much saw like there were differences in cortisol levels between like the night and day times, where it helped with increased regeneration. I believe it was at night, but I'm not sure. I think one thing that you can also probably look into is GABA. They have done studies where they're not looking at like day and light and night, but they were looking at if you were to disrupt like the SEN, if it would lead to changes in GABA receptors. And pretty much, I believe they did see some differences, but it would be interesting to see in this.
Yeah, totally. I mean, like I said, we have serum banked. So I think doing kind of this metabolite analysis or looking at different hormone levels and stuff would be super interesting. And I'm positive we would see differences based off of time of day, and especially things like cortisol, which is known to have a time of day dependent effect on its secretion. So I'm sure we would see a difference if we looked at it. And we have the serum, we have tissue banked to look at all of that stuff.
Can I ask you another question? So this is probably more of a, I want to see you think and not necessarily based on your data, but if you have these results, this is mostly to see if a PhD student can defend themselves. So if you have these results which show no difference between light and dark, why are you bothering to look for a clock? Why are you looking at gene expression of your circadian rhythm proteins? If there doesn't seem to be any light dark especially specific, what's the rationale for? And that's not to say it's a bad experiment. That's not at all what I'm saying. I want to hear your rationale for why, after seeing these results, you're gonna still study those genes.
Yeah. So my rationale here is, so comparing this paper to the papers that I have a problem with, where they do, in my opinion, incorrectly done exercise tests and then look at, whatever, core clock gene transcription or something like that, and show that it changes based off of time of day, and then make that correlation of, oh, look, this is changing based off of time of day, and this performance on this test is changing based off of time of day. This must be why we're seeing the effect. I think that is an inappropriate jump, and I think to show that, I need to show that even while these things are changing based off of time of day still, for example, core clock genes, like BMAL1, CRY, PER, that even though that they are changing based off of time of day, when you actually look at the muscle and test it in this way where you're controlling for all these different variables, you don't see an effect. Because if we do this test and we don't show a time of day difference in different circadian genes, someone could argue, oh, well, you're just inherent to your experimental design, you're just ablating circadian rhythm, and that's why you don't see an effect, because you're removing the muscle and you're doing it's ex vivo and it's electrical stimulation. So, someone could say, oh, the core clock proteins are being degraded during this experiment, and that's the only reason why you see any differences. So we want to actually measure these things that have been known to vary based off of time of day. And we have preliminary data showing that even in these samples that they do vary based off of time of day. And even when the circadian landscape and transcriptomic landscape is completely different within these different tissues, when you actually test it in this fashion, the physiology, how it's actually performing on these tests, doesn't change. And I just think we need to measure those circadian proteins, for example, to show that this is real and the effects that we're seeing aren't just due to ablation of the circadian cycle.
Cool. You should lead those future experiments with that rationale. That's solid.
Gotcha. That is good feedback. Thank you. And so, as I mentioned earlier, we not only measured muscle contractile properties, we also measured muscle glucose uptake. And so we did this with the radioactive 2-deoxyglucose approach. And so for this experiment, we take the muscles out, and one soleus and one EDL is rested, and one soleus and one EDL is contracted. So these are the muscles that we got the data from on the slides earlier. So they go through the contraction protocol where it's a force frequency curve, and then an endurance test. And then after that, or during that, one either is rested and gets incubated in the radiolabeled glucose, or contracted and bathed in the radiolabeled glucose. And this allows us to, because the glucose is radiolabeled, we can track how much glucose is moving inside the muscle, either during the 15 minutes of rest or the 15 minutes of contraction. And this is what the data looks like. So this is just looking at one time point. So we have looking at rested versus contracted muscle. This is at Zeitgeber time 1. And the y-axis, we have 2-deoxyglucose uptake. And so you can see that when you compare the rested to the contracted muscle, the contracted muscle takes up more glucose. And one way that we can quantify that is C-stim 2-deoxyglucose uptake. And so we get this value by subtracting the rested value and the contracted value for each mouse. And the difference between the two, we say, is the contraction stimulated glucose uptake. So that's another way of looking at this data. And no matter what time point you look at, whether it's ZT1, 7, 13, 19, there's no difference between the groups in glucose uptake or contraction stimulated glucose uptake. And this is the case not only for EDL, the more fast twitch muscle, it's also the case for the soleus, where irrespective of time of day, there's no difference in glucose uptake between the different muscles. And so kind of just the main takeaways that I want people to walk away from this talk with is that the circadian rhythm has a molecular basis. There's an actual molecular clock, which I think is just really cool and really fascinating. I mean, I remember even as a kid waking up before my alarm clock, a minute before it's going to go off. I'm wondering, how do I know that it's 7 a.m? And just the fact that there's an actual molecular, mechanism for it, I just think is just so unbelievably interesting. So that's main takeaway number 1. Number 2 is that there's a documented time of day effect on athletic performance. However, in my opinion, most of these studies are poorly controlled and erroneously attribute time of day effects to endogenous muscle changes. And that our data suggests that these time of day effects are not due to endogenous changes inside the muscle, and probably more likely due to factors external to the muscle itself. And as I talked about earlier, I can say that with confidence because our studies controlled for many different variables that other studies do not control for in the field of chronobiology. And when we do that, we see no difference in force production, fatigability, or glucose uptake in response to the contraction protocol. And so that's all I have for today. I think I'm just about at time. Just like to say thank you to everyone in the Shank Lab, my advisor, Simon Shank. The Ward Lab has also been really helpful with this as well. So specifically, Shannon, she built and created the ex vivo contraction chamber. And if I didn't have that, I wouldn't have this project. So I cannot express my gratitude enough to Shannon for that. And Dr. Yoshi Cho, who's going to be helping out with some of my molecular analysis down the line. And finally, last but not least, I'd like to thank everybody in the audience for attending and listening and asking great questions. I really appreciate it.
Thank you very much. That was really great. I really enjoyed it. And it's very interesting to really see, how all this previous work, did not consider there might be it's not a clean system for studying this, and getting a better clean control system can, rule out some of these conclusions that they have. So do you plan to compare your ex vivo results with monitoring the muscle function and contractibility in vivo directly with the same design? So then you can see that, well, when you don't control this, you don't take the muscle and evaluate things far from all these things that can affect, you really don't see effect directly in the muscle. But if you have the whole context, then you see it, meaning that it's these other parameters that are affecting biologically the function of the muscle. What are the response that the previous studies have shown?
Yeah, I mean, one thing that we've even thought about doing that I think would be interesting too is kind of taking something more in the middle between ex vivo and in vivo and looking at things at a in situ approach. So stimulating the muscle, inserting electrodes into the leg of the mouse and not surgically removing the muscle and stimulating them that way. And then measuring force production. I think that'd be really cool because you can control for a lot more variables, but you still, the muscle is still innervated, the neuromuscular junction is still intact, it's still receiving those humoral factors, and we can still standardize stimulation and take out the effects. The mouse is anesthetized, so we're taking out differences in pain tolerance or perceived fatigue. We can still do a standardized last meal. So that's an experiment I think would be really interesting. I just have to learn how to do in situ contractions first.
Good. Is there any other question?
I had a quick one. So
Yeah, go ahead.
I'm glad you've collected serum samples. I Liz beat me to asking about cortisol, but so I think cortisol, and I don't, you guys know more about this than me, is involved in the glucose uptake cycle as well. Is there resident cortisol that would actually influence these things in ex vivo? Or is this something which is more systemic? What's the I guess the timeline and the relationship of those interactions as they would influence your ex vivo data? I don't know if that question made sense. I guess the short version of that question is, do you have cortisol which might be affecting your muscle outcomes ex vivo?
Yeah, that's a good question. I don't know the differences in, serum concentration and cortisol based off of time of day, but
I guess take out the serum concentrations, because those serum concentrations are not really directly affecting your ex vivo results. Is there, like, if you explant a muscle, is there cortisol in that muscle, and does it play any role, or does it have to be like does it have to be a systemic effect as it relates to metabolic activity? I don't know the answer to this question. It might be a question for Simon.
I also don't know the answer to the question, like how much cortisol is typically in, a muscle and how much that changes based off of time of day. Yeah, I'm not sure of the answer to that question.
Can you measure cortisol in your muscle samples? A muscle lysate?
That's a good, I mean, probably. I'm not sure though.
Yeah, we can talk about this later. I think time's up anyway. I don't want to drag this on for anyone else, but I'm glad to hear that the nervous system still matters to all you muscle people. That's all I'll say.
Okay. If there is no more question, thank you very much, Liam. Great, and I'll see you guys next week. Thank you.
Bye.
Thank you, Liam.
Yes, thank you.
Have a good day.
Thank you too. Bye.